Good morning, everyone. Are we ready for that break tomorrow? Yes. Okay. I'm a bit surprised. I thought it's more students than I thought I'll have the day before Thanksgiving break. How about we do this? Because you came here, I'll just give you one, two, maybe three questions from the final exam, but you don't tell anyone else that came in. How about this, right? That would be great. I didn't write the final exam though, so I cannot really do that, but if I had, I probably would have done that. Not really, but still. Okay, we guys are entering our very, very, very final system, 12 systems. We are about to enter the last two lectures, two class meetings, talking about the reproductive system. This is going to be not scratching the surface, going a bit deeper than the surface, but again, because of time constraints and so on, it will be to some extent. I think with the female reproductive system, from my experience, we will learn about some things that you do not necessarily know. But it might be more of a review for most of you in many cases, and we're good with that. Okay. So the birds and the bees that we're going to talk about. So what I'm going to talk first of all, big picture. We're talking about the male and the female, and we're going to start with the male reproductive system. Today we'll complete, hopefully, the male reproductive system. We might just start thinking about the female and do some compare and contrast. We'll see a lot of similarities, but also a lot of differences. And then after Thanksgiving, in our final class meetings, we'll talk about the female reproductive system. We will not have the time to talk about all this part of what happens after, development and so on. Many of you will learn more about this in development biology, but unfortunately we don't have the time. But the textbook is doing a pretty good job on the development of the fetus, the embryo, and so on. So just for general education, I guess, I encourage you to read that. Okay, so we'll start with the male reproductive system. The overview, what we're going to talk about first about the major reproductive organs, the testes, or testes in plural. We'll talk about a bit about the anatomy and a lot about the physiology of the testes. We'll talk about the formation of a sperm, of a mature sperm, which is basically two processes. One is meiosis, and the other one is what we call spermiogenesis. Altogether, we call them spermatogenesis, genesis, creation of, the formation of a sperm. We'll talk about the semen, and this is not much that we're going to talk, but it's mostly based on what you read, so you are familiar with the semen, so we'll just mention this, but it's essential. And then we'll go back and talk about all of this, but now we'll learn about reproduction. We'll see how we're going to go back to concepts that we learned way, way, way back. And we'll talk about the pituitary gland, and we talked about the hypothalamus and the axis. We talked about the adrenal glands, and we talked about the thyroid glands. We're going to use the same concepts, but now talk about the testes. And then finally, we'll talk as much time as we have about sexual behavior, the male sexual behavior. Okay? So let's start. And I'm going to start with a question that there are so many of you that, if not asked by now, you're going to ask it at some point in your life. And it's a question that makes a lot of sense. What do we actually need them for, right? What do we need them for, guys? And when I say guys, I mean the guys in the everyone context. What do they contribute to the whole thing? So they contribute sperm for sure, but what do they contribute with that fertilization thing? What does the sperm, do they bring the same thing, the oocyte from the female and the sperm from the male? No. What do they bring? No. Pure mentoring genetics, chromosomes, that's what they bring. They bring half of the genetic material, not much more than that. A little bit more than we thought they do, but that's basically it. I do want to say something about today and the final class meeting, just to make sure we're not missing anything. We're going to talk about today about human reproductive system. We're going to talk about males and female. I'll say male and female a lot, right? When I say male and female, I'm talking about male and female. Not about men, not about women, not about gender. Okay, any gender whatsoever is more than welcome in my class. I want to make sure we're clear with that. We're talking about female, we talk about XX. We're talking about male, we're talking about XY. simply, just the physics of that, or the physical property of that, okay? So, yes, that's what contributes mainly, half of the genes of the progeny. A bit more than that, but that's it, which is very different compared to what the female brings, right? And we'll talk about the female. That's one of the compare and contrast things. So, the functions of the male reproductive systems are the following three. First of all is the actual formation of the gametes, of the sperm in this case. Then, synthesis of the male hormones. We call them in general androgens, and we have one that is maybe more important than the other. Which one is that? Testosterone and its derivative, but we'll talk about testosterone a lot. And then finally, the male reproductive system is also important for delivery of the sperm, because the sperm is made in the testis, and it needs to go all the way out, eventually to reach the female reproductive system. So we need to make sure it can go through all the ducts and such. And extremely important, as we will see later on, it also needs to travel later on in the female reproductive duct. Okay? So we want to make sure, think about someone who's going for a journey and brings a backpack with it, right? And needs a lot of things like energy and other things to survive in that different foreign environment. So that's the roles of the function. And just as a very big gross anatomy, we have external genitalia, the scrotum, basically the layer that covers the testis and the penis. We have the internal genitalia, the testes, that although they are external, they're also in a way internal because they are covered, and all the different ducts. And we have the accessory glands that are responsible for making what as a whole? The semen. Exactly. Also essential, we have three parts. We have the seminal vesicles. Right here we have the prostate, and we have the bulbourethral gland, which by itself is not essential, but it helps. And we will briefly talk about what they contribute. You are responsible for them at the level of the GRQs/assigned beings. Okay? So we're going to focus first on the major factory of sperm, the testis, right? Anyone know what we call the testis body? Testis sounds familiar, sounds from what? Testosterone. It's actually, testosterone will come from the testis, but where's the testis coming from? Sounds like another word in English. It's actually coming from Latin, but originally from English. Something really. I'm testifying or testimonial, right? Sounds good. And that's actually the origin for that. How come? Back in the days in the Roman Empire, so today, for example, if you go and testify in court, okay, what do you do before you start your testimony? Right hand here, right? Left hand here, and you swear, right? What do you put your right hand on? The Bible. In the days of the Roman Empire, the Bible was not a bestseller, to say the least, right? So instead, they put it somewhere else. Where somewhere else? The only people that were allowed to testify are only males or men at that time. Okay? Women were not allowed to testify. So in a way, what they put, and I'm not going to demonstrate, is they put it on the place that shows that they are men in a way that I am allowed to testify. So basically that's where it's coming from. It's from testimony, going to Latin, and eventually going to English. Okay, so our testes are internal, as I mentioned, but to some extent they are external, right? They are outside. Now, until now, we talked about 11 systems. All of them are essential to the individual that carries them, right? Each of us needs a digestive system. Each of us have to have the respiratory system, and so on and so forth. For our individual part, do we have to have the reproductive system? That's the only system we can deal without, right? We can define. In other perspectives, right, that's the most important system that we have. Evolutionary-wise, you don't have a reproductive system, you're just an individual, that's it. You don't have a species for that. So we're taking a most precious thing, evolutionary-wise, and during evolution, we put it outside, the most fragile place you can imagine, right? And any male who experiences it whatsoever, right, some getting the right the wrong heat in the wrong place knows how precious and how bad it is when it's external. So, and it's not accidentally there, because when you're an embryo, you guys know where the organ starts? It's in the abdominal cavity, up there. So it descends actively and going down during pregnancy until it makes it out. So that's one of the things that we ask ourselves in evolution, what's the advantage if there is some advantage, okay? Anyone knows? What will be your first answer based on what you know about what do we gain from the fact that it's external? Temperature. Why is temperature? Sperm cannot tolerate 37 degrees, which is centigrade, which is our body temperature. The sperm will die, okay? So we had a solution for that during evolution. We just take it out of the system, right? Can you think of a better solution, maybe? Think about it a little bit. Any creative ideas? Malik? Maybe you can just have evolved sperm that can tolerate 37 degrees. That would be so much easier, right? But this is not the way it evolved. And there are many ways by which we can explain, but one of the things that are very important about evolution, for those of you who have not studied evolution, right? It's not that everything that we see today is necessarily making sense or provided any sort of advantage. It at least did not provide any disadvantage, therefore it survived. But many of our processes have basically evolved. It's not just natural selection, it's not the only mechanism, but just evolved because they were okay. Some other people will suggest, some other evolutionists will suggest that maybe it isn't the most fragile place to actually show that you are the male, the best, the fittest, because here you can expose your most precious thing and in a fragile place, basically. So you can choose me, the male, I'm the fetus. And maybe that's the explanation. But just something to note. Okay. So when we look at the testis and we just remove part of it so we can see, that reminds us of something else, right? What else do we learn about that looks exactly like that, basically? This is exactly the kidney, right? And the nephrons and everything, different names and so on. But basically the same idea, with a core, with something that looks like medulla, it's not a medulla, and with the tubules inside that are highly, highly, highly folded. Okay? Concept that works where once can work in another place, in a different organ, in the same idea. So here we're talking about tubules that are called the seminiferous tubules, and these are our factory of sperm. And if we make a cut and rotate, that's basically what we will look at an electron micrograph. The colors are pseudo-colors through the computer, but the image itself is real. And what we'll see is the sperm factory. And in general, we are starting with a precursor here and develop or mature the sperm all the way as they go inside into the lumen. And here you'll have the spermatocyte, eventually spermatids, that will run in the lumen and eventually make it out. Okay? So let's talk about the process of making the sperm. And stop me with any questions that you might have. As I told you, the last lectures, I'm rushing a bit and less activities for the sake of time, to make sure we cover it, but if I'm rushing too much, tell me. So we're starting with the stem cells. What does it mean when I say stem cells, and why do we need stem cells? As a whole, I'm not just talking about the sperm. It can be the muscle. Why do we need stem cells? What are they? How are they different from other cells? They don't quit dividing, exactly. Our normal cells divide once or twice or 50 times, up to 50, depends on the type of cell, but at some point they reach what we call senescence. They cannot divide. They're fine, they're healthy, they do whatever they do. They can be skin cells or muscle cells, but they cannot divide anymore. And for that we need stem cells. These are not the embryonic stem cells. We're talking about somatic stem cells in each type of tissue. In this case, these are the stem cells that will give rise to eventually to sperm. Okay? They're found in the periphery. They're called the spermatogonia, spermatogonium in singular, yum and ya in plural. They reside at the periphery of the seminiferous tubules, undergoing mitosis, simple mitosis. The more the better. Yes, Sierra? We call them germ cells at this point, and we can put them as the germ cells, but they're behaving just like regular somatic stem cells, not just somatic cells? Good question. Okay? And then they go mitosis, nothing unique, right? Until a big event is coming, and that big event is, when are things going to change in the life of that young male? At puberty, exactly. 12, 13 or so, somewhere like that. Bar mitzvah just came, and the guy gets some hormonal secretions, and we come back to the hormones, right? And now what we have is a change, and part of the change is to make these unique, exceptional cells that we're going to talk about. So upon puberty, the spermatogonium, in singular, spermatogonium in plural, will continue to give rise to oligomitosis, but now there will be two different cells. Well, typically it was just another cell of spermatogonia. Now one of them will be spermatogonium, because we still need the stem cells, right? Second one is a unique cell. It's called a primary spermatocyte. It's the only cell in our body, in the male's body, that can undergo meiosis. None of the other cells will be able to do that. It's also unique in the sense, and I'm not going into developmental biology here, but it's a cell that carries, in a way, all the developmental information, developmental plan, if you will, of what will become an embryo and a human being later on, which is very different from all the other cells. Okay? All the genes are there, all the genes can potentially be expressed, which is very different between different tissues. So I do not require you, you're not responsible for learning all the different processes and stages of meiosis, right? Just remember that basically you learn meiosis, or at least in Bio 101, right? And we know two meiotic divisions, okay? But the basic things I do want you to know is about the basic two meiotic divisions and just understand the numbers. So let's see what we hear. This is the periphery, this is the core, this is the lumen, right? We start with a spermatogonium. We're diploids, we have 23 chromosomes and the two sets of 23 chromosomes, right? One from dad, one from mom. So we have 46 chromosomes. And this is mitotic division, but at some point, we'll have division to a daughter cell and a primary spermatocyte. And this is where meiosis begins. So primary spermatocyte undergoes meiosis by two successive divisions. The first one, it splits into a secondary spermatocyte, which carries now half of the chromosomes. And finally, the second division, which will be formation of fourth, and we call them spermatids. Almost mature sperm. Meiotically, at least, gene number, chromosome numbers, we're good, right? So let's make sure we got the numbers right. Let's talk about the secondary spermatid. Okay, spermatocyte. Let's talk about a gene, specific gene. Give me a name of a gene. Gene someone is working on in the research. Come on, Diego, throw me a gene. SOX9, wonderful. So SOX9 is a gene, like many other genes, we have two copies, right? One we got from dad, one we got from mom. I want you to think together, and then I'll count to three. This should be short and quick, okay? In this one, in the left spermatocyte, how many copies of SOX9 do I have? Again, in the left one here, how many copies of the secondary spermatocytes I have? You ready? Ready? 1, 2, 3. And we are all wrong. Why? Because we have sister chromatids. Because we have 46 chromosomes, but before meiosis took place, we had duplication of our DNA. So we actually have 92 chromatids, right? Each chromosome has 2 chromatids basically, so 2 copies of each. The paternal one we got from dad, is now 2 copies, and the maternal one has 2 copies. And then in the first division, what we do, we separate the homologous chromosome, the paternal from the maternal. We have 23 chromosomes, but how many chromatids at this point? 46. In the second division, this is when the sister chromatids divide, and now we have single chromosomes and single chromatids, and now we have just 1 copy of the SOX9 gene. Okay, any questions? Don't feel bad. This is the 10th year I'm teaching that, and every semester everybody's wrong, so you're good. This is just to remind you to go back, just make sure you understand the numbers, okay? Question? Okay, so we're good, right? We have spermatids, and each one of them carries half. Why do we need just half? Why not just keep them diploids? What's going to happen later? Guys, help me. Fertilization, and we're going to make the oocyte, right? And we don't want too many copies, we just want two. So the spermatid will carry half, and the oocyte will carry half, and we're happy. So are we done with spermatogenesis? We have a cell, it has the right number. Are we done, or do we need to do anything else? Guys, help me. They're not mature in what sense? What do they need to undergo? I mean, they're done with meiosis, they're good with the numbers. Why aren't they mature? Why aren't just going and doing the thing? Why to invest more energy in more processes? For example, to be able to travel, right? They are there in the testis, it's out there in the penis when they need to leave. And again, remind you, they need to reach not just to reach the female reproductive system, but to make it through and to survive. No offense to the females, but we're talking about a harsh environment for the naive and plain young sperm. It's a very hostile country, from acidity to many other things that they will need to travel with, right? Who saw Lord of the Rings or read Lord of the Rings even better? Just that? Come on, raise your hands. Okay? So remember the backpack, they're going to Mordor. Remember that? But they have, right, they have those, I don't even remember how they call the biscuits, cookies, how do we call them? Right, the bread, the special bread that they take. They need some packaging, they need some equipment because they go into that hostile country, they need to make sure. Okay, from the sperm perspective, female reproductive system, no offense to anyone here, Mordor, completely, right? So let's see what we help them with. So the second part of the whole spermatogenesis is what we call let's prepare them. And that's called spermiogenesis. We're changing the shape, we're reorganizing the sperm. The first thing that we see is the formation of a vesicle that what we'll continue to develop, and we call it the acrosomal vesicle. It contains acrosomal enzymes, and they are going to do what later on? They're going to move and help penetrate through the protective layers of the egg, of the oocyte. If you are not a good sperm in the sense of equipped sperm, but in your backpack you have enough acrosomes, you're not going to make it. Others will compete, others will win. It continues to grow. This is the one that you see here in the red here. But at some point, the other thing we see is the formation of flagellum. Many of our bacteria have flagellum, right? And this allows them to move. They have motor, they have ATP pumps, and they can create energy and move. It's by itself, as you will see, it's going to be pushed, but that swimming part will help that sperm make it. You're swimming better, you're better fit. You might reach and provide your genes to the next generation better than your other friends, your other peer sperm. Okay? The other thing that we see already here is that a change in the cytoplasm itself. The nucleus is getting very condensed. The nucleus is right here in purple, and it contains that precious gift that we're going to provide the embryo, which is what? We talked about that. It's the genes, right? The chromosome. That's what we need from the sperm. But we need a lean, mean, running machine, and we are going to lose any excess weight, including all the cytoplasm and the fluid and so on. The other thing that happens as we are losing part of the cytoplasm, we're still keeping mitochondria, but not only we're keeping, we're recruiting, accumulating more of them next to the nucleus. What do we need that for? In one word? Energy, and eventually ATP. Great. In order to utilize ATP, we talked a bit about that in the digestive system, right? We need a source. So one of the things is mitochondria that will provide us the ATP, but what's the source for the ATP? Anyone remembers? Fructose. If you remember, we talked about that. That's the only cell in our body that can actually utilize fructose directly. And fructose we're going to find in the semen itself. And finally, we release the exit cytoplasm, and look at what we have here. The swimmer is ready for the journey. A lean, mean, fertilizing machine. Are we ready yet? No, not yet. In the testis, we're not yet fully mature. We're actually not yet fully motile. We need to do some other things to be successful. The first explanation is a scientific one, and then we'll talk about some others. What makes you win? Just once more, I like it. Stop it here. Flowers never hurt. So, but it is true. Scientifically, it is true that they're not yet ready. They're not motile, they have a flagellum. And when they are more like, not like, but they are more pushed with the flow. Remember, we are making more and we're making more and we're making more, and this pushes the ones that are made inside the ducts, okay? And they're going to the next step, the next duct, which is the epididymis, and this is where they are getting secretions of cells in the walls of the epididymis that eventually make them fertile, or make them mature enough. And this will be about 2 months, 2.5 months. So it takes some time. Okay? At the end, by the way, at the end of the epididymis, they're going to sit and wait until when? And I'm not going to go into the ducts. All you need to know in the ducts is at the level of what you read. Okay? Until when I'm going to sit at the end of the epididymis? What's the next duct? Right, you read. It's the vas deferens, right? And for me, we go into the duct, spinosum outside. So until when it's going to sit on the end of the epididymis, it looks like a U-turn. Anyone? Ejaculation, exactly. It's going to sit there, right? Until it ejaculates, whether it's a sexual behavior or not a sexual behavior, it doesn't matter, right? We're going to leave. Now, this can happen in quite a while, right? As we all know, it doesn't always happen, that sexual behavior thing. So, isn't it a problem that sperm is going to wait and wait and wait, undergo different chromosomal aberrations and so on, and older sperm is more susceptible to mutations and so, right? And the answer is true, but it's not really a problem. Can anyone think why it's not a problem? How we solve this problem? Yolanda. It's going to be reabsorbed, it's going to die and reabsorb, it's going to undergo apoptosis. But it's not a problem, because we're creating 300 million of these the next time. And males are, if everything is okay, they continue to be fertile. It's a factory that goes on all the time, so it's not a big deal. You just make a lot, knowing that most of them are going to die. Okay. Before we move and talk more about hormones, I want to come back to the seminiferous tubules and remind ourselves that it's first and foremost, we have the germ cells, right, the sperm in the different stages of development, but we also have two groups of somatic cells that are critical and essential. So in this diagram here is the lumen. Blue, you see all the germ cells, the different sperms, spermatocytes, spermatids, etc. But we also have two groups of cells. One group is actually outside of the seminiferous tubules. They're called the Leydig cells, after Leydig. The scientists discovered them. And then we have big cells, large cells in between those groups of germ cells, and they're called Sertoli cells. If we zoom in, okay, in this case the Sertoli cells are in yellow, so here's one Sertoli cell, and here's another Sertoli cell. You can see the membrane between the Sertoli cells, and they are enwrapping all the developing sperm cells, right? Starting from spermatozoa all the way to spermatoids. What is the role of these Sertoli cells? Let's think about a potential problem that we have had if we didn't have Sertoli cells. Now, outside here, you have interstitial fluid and oxygen, amino acids, everything that we discussed, like any other interstitial fluid, right? but the capillaries don't make it to the cells themselves. So these spermatozoa, how will they get fed with nutrients? What's the process that works here? I need to hear it from all the class. We're at the end of the semester, hopefully. We got something. Again, we don't have capillaries that go to the cell and say, here are your nutrients. They go here, they're around my laser point right now. So what's the process? 1, 2, 3. Diffusion. Thank you. I'm scared for a little bit. Okay, so diffusion, right? Which is great. The problem is, they're really close. But as we go and develop further and further and further away, we have those spermatids at the end that are really in distance from the nutrients. So diffusion cannot solve this problem. What do we have? We have function follows form, an anatomical solution for that. We have these large cells that a single cell will start where diffusion provides the nutrients all the way to the end of the spermiogenesis form. Or spermiogenesis ends, all the development, right? And spermatogenesis ends. So the same cell, these will represent the nuclei, the purple, gets its nutrients, let's say, for example, amino acids. But these amino acids are going to make it to all the cell and then provide those nutrients to the developing sperm. Does it make sense? So they serve as what we call nurse cells or nursing cells. We typically call them nurse cells. Any questions about that? There's another interesting phenomenon, and this is something I want you to think about together. So we have this membrane between the cells, right? And these cells, as always, have some pi junctions. We talked a lot, right? And we learned that sometimes it can be leaky and sometimes it can be not leaky. And sometimes it can be, we talk about an example where there's no leak, right? There's a huge concentration of tight junctions and therefore it's all sealed. How do we call that place? The BBB, right? When we don't want anything to move. The same thing actually goes here. We don't call it the BBB, we call it the blood-testis barrier. What does it mean? It means that if the capillaries are right here, we don't see them, nothing can really leak from the capillary. Things will leak from the capillary to the testis, but nothing can really, to the interstitial fluid, but nothing can leak in between. So nothing from the blood will leak in between these because everything is full with tight junctions. The only place we can get nutrients from and get rid of waste is going to be through the Sertoli cells. Now I want you guys to think together for a second, okay? It has something to do with something we learned in the past. Why would we need a blood-testis barrier? What's the logic? What could have happened if we didn't? Let's think together. Okay, all of them were wrong, by the way, but it's you cannot tell, right, if you don't know, so it's fine. Anyone wants to suggest something? Yes? Okay, so we'll talk about that. Just leave it for a second, okay? We'll come back to this. Anything else? Yes? Yeah.
We were thinking that an infection, if it were to reach these cells, since they're constantly dividing, it could spread.
Wonderful. That's one of the comments that I got. It's not that it's wrong. It's actually helpless because in the sense that we are, we're talking about in the brain, we talk about fragile cells that we don't want to risk, definitely for our sperm, right? We don't want to have viruses, bacteria, other pathogens going there. And it's not that it's wrong, it's definitely one of the solutions to take care of that. It's not going to be the main answer. Another question that I heard, another answer that I heard, was maybe about the temperature, right? We talked about the sensitivity. Now, the blood carries temperature. That's where the temperature comes, right? The energy, the heat, maybe it's too close. So that's an interesting point that we haven't discussed earlier on, that actually the blood vessels, the capillaries in the testis as a whole, including those that will reach that seminiferous tubules, are actually in the right temperature already. This is not for the exam, but just an interesting thing. Kind of like based on the same concept of the kidney and the loops of Henle, the arteries and arterioles that go into the testis are meeting as they go in with the veins coming back from the testis. The veins come with what? Cold or hot temperature? cold, relatively cold, right? And going down, you're coming with a hot temperature, warm temperature. What happens in exchange of energy? The veins become hotter as the arteries become colder, so they reach testis when they are about 35 centigrade. So actually, we don't have a problem with temperature. Isabel? So after that, that would be a mature sperm. Isabel is asking about the female reproductive system would be good. Now we're done. It's more about the formation, the stages and development towards that. So I'm going to go back and talk about the immune system because, again, it's helpful that we are fighting pathogens, but there's another thing we need to fight with or need to be very careful about. When we learn about the immune system, what did we learn at the end of our immune system, the really last part? You remember how we call that process? Thymic education, wonderful. What happens in the thymus? Who wants to remind us?
You have cells in the thymus that are basically present autoly active.
So we have in the thymocytes, the cells in the thymus that say, hey, here is my antigens, my peptides, right? And what do we do with T cells, and the same thing goes for the bone marrow and B cells, that do recognize our own self-antigens? What do we do with them? Do we want them? No, right? They're bad. They're going to attack our cells. So we kill them, right? We trigger a process of apoptosis. So we got rid of that. All of this takes place when we are embryos and we are very young infants, in the very first months, right? When did this process start? When did it start? Sorry. At what age did we start with all the spermatogenesis? After puberty. You're 12, you're 13, your immune system is there, right? So basically, we're going to have new cells, and with their new function, these are very unique cells like sperm, and they're going to express some proteins that other cells do not. They were not there in the thymic education. When our immune system was educated and when we filtered and selected against those that know ourselves, we did not show them the sperm proteins because we didn't have sperm at the time. So exposure, in this case, might lead to the sperm being attacked by our own immune system, being recognized as a foreign tissue, as foreign cells, foreign antigens. And therefore, we make a clear separation. That's the blood testis. Yes, Miranda.
Is there a common mutation that results in fertility and
I don't know if there's a common mutation about that, but we sometimes have exposure as part of diseases, and that might lead to infertility. Exactly. So these are the Sertoli cells and Leydig cells, and they have two very important functions other than that, and that's in making and helping with hormonal regulation. And that's the point, to go back and see how we control, how do we tell the body at puberty start to do all of these meiosis things, spermatocytes and so on. And this comes back to the endocrine system, and here's the thalamus, and here's the hypothalamus. We don't care about the thalamus in this regard, the hypothalamus and the pituitary gland. So we have the brain testicular axis, just like we had the brain thyroid axis. It all starts where? Where's the CEO of all this axis? Hypothalamus, right? So we have hypothalamic proteins. We call them in total, as a group, the gonadotropin releasing hormones, GnRH. And they're going to which pituitary? Anterior or posterior? Let's see that we remember something. Anterior or posterior? Anterior.